how do you call it, governmental and private agencies who have contributed to cancer research in Ghana. So one, is there a database available for researchers to study trends and guide therapeutic interventions and innovations? Is there sufficient collaboration between researchers and clinicians in advancing cancer research in Ghana? Also, are there policies informing data collection? Are there funding opportunities for localized research for students interested in cancer research? Also, are there programs, short courses, and training for cancer immunology, cancer genetics, radiation oncology, nanomedicine, etc., for medical professionals who are interested in research? So, again, at ACRI, our focus is not to compete with any established cancer center or industry player. We seek to advance cancer research in Ghana through awareness creation, creating collaborative teams of students and all professionals interested in cancer research with the goal of generating locally relevant data and strengthening research capacity. Also leveraging local expertise and resources to address present cancer changes, in terms of funding in our local communities by prioritizing projects that are feasible yet impactful and collaborating with industry players at the highest level to gain skills and access to data for high-end research. And so we have brought about our ACRI Expert Series. As you all know, there's an expert series to create a platform for knowledge exchange between global cancer researchers and biomedical scientists. Also bridge the gap between basic scientific discoveries and clinical application, as well as showcase emerging technologies and innovative approaches while transforming cancer research, inspiring collaboration among researchers, clinicians, students, and healthcare professionals, and building research capacity by providing participants to cutting-edge development in cancer science. So we've had two sessions. The first one was on cancer immunology and cancer neuroscience. The second one was artificial intelligence and nanomedicine. And today we'll be having the final session of the second and quarter edition of our series. And so today's topic is immunotherapeutic approaches in sarcoma management, the emerging target and translational insight. So the presenter will seek to focus on the evolving role of immunotherapy in the management of sarcomas and other rare malignancies, emerging immune and molecular targets with potential therapeutic significance in sarcoma treatment. Also, advances in translational research driving the development of novel immunotherapeutic strategies, challenges and opportunities in translating preclinical discoveries into effective clinical interventions for sarcoma patients, and future perspectives on precision immuno-oncology and combination therapies for improved patient outcomes. So today we have a wonderful presenter. She actually was my teaching assistant. She mafiaed me small. She's in the person of Augustina Quarshie. She's a PhD student, a molecular biology PhD student at the Institute of Regenerative Medicine, the first Moscow State University, Sechenov, Moscow in Russia. So, permit me to talk briefly about Ifia. She is a biomedical scientist by profession and a researcher at the Institute of Regenerative Medicine at Sechenov University in Russia, and working within the Laboratory of Molecular and Cellular Tumor Treatment at the University's Institute for Regenerative Medicine. Her specialized work focuses on oncology, tumor environment, and finding innovative targeted treatment for aggressive cancers. So that is who our presenter is, and I believe that she's going to give us a wonderful presentation. And so please sit back, enjoy, ask questions, challenge ideas, and let this platform shape your curiosity and ambition. Thank you very much. So, Ms. Augustina, please, over to you. Thank you.
Thank you so much, Orwell. Okay, so I wouldn't waste much of your time here. We will just go straight to the point and then begin. Can I share my screen, or you have to do it for me?
Oh, please do share. Please do share.
Okay. Okay. Please, I hope everyone can see it.
You can.
Okay. All right. So we will just start. At the end of the session, I will switch on my camera, just not to distract you. Okay. So please bear with me. So today we will look at immunotherapy. What even brought about immunotherapy? How did we arrive at immunotherapy? The short background story is that scientists tried surgery. I mean, it was the major point of treatment of all cancers. So they were stuck to surgery, and then chemotherapy also came because they realized that after the excision of some cancers, there are still remnants. There are still some cancer cells that are not absolutely eliminated. And so they decided to use chemotherapy to eliminate the rest of these cancer cells. And then after that, they tried again and they realized that people were still getting relapse. After the cancer has been excised, after chemotherapy has been tried, people were still presenting with relapse. And this was when immunotherapy came in. They realized that the body has its own mechanism of fighting infections. Whichever foreign antigen that is introduced into the body has its own mechanism of eliminating it. And so they sat down and said that why not emphasize on these cells, on these immunocells that naturally takes away foreign antigens? So that was when immunotherapy became the highlight. So the table of contents, we will look at what cancers are. We will look at cancer immunotherapy. And then we will break down to sarcomas. What are they? We will also look at osteosarcoma, which is my field, what I'm doing right now. And then we will look at the molecular perspective of osteosarcoma. We will just go to the tumor microenvironment when there is an osteosarcoma, what is actually within this cancer. We will look at the cure for osteosarcoma, what the cure that started and how we have arrived. We will also focus on current research. We will look at my review paper and then my research, which I'm doing currently. So cancers, we classify them according to the cells they originate from. So scientists looked at which cell is actually which cell actually presents a mutation, which cell is malfunctioning, and then they use that to classify cancers. So we see carcinomas that originate from epithelial cells. We see sarcomas that originate from mesenchymal cells, lymphomas and leukemias, which are blood-forming cancers from the bone marrow. Carcinomas are the major form of cancers because every tissue or organ in the body has epithelial cells. And this type of cancer is mostly seen in adults. They are seen in adults because, you see, as you age, you are exposed to a lot of mutants. And so these cells, as they accumulate these mutants, the genes in these cells undergo mutations, and you end up with the cancer. We have prostate, breast cancer, which is very common. We have prostate cancer, we have lung cancer, pancreatic cancer, and then colon cancer. Sarcoma, previously it was not diagnosed because it was very rare. It is a rare type of cancer. So previously the attention was on carcinomas, but we evolved to a stage that people just go to the hospital and say, I want to run a full body checkup. And that was when they started discovering the sarcoma, which happens in mesenchymal cells. And It develops from connective tissues. These mesenchymal cells are within the connective tissues, like the cartilage, the bone, fat, and muscles. So this is where this type of cancer arises from. Lymphoma and leukemia as from the bone marrow. So what is unique about this cancer is that immunotherapy has been effective because of the liquid nature and how these immune cells are able to circulate in the blood system. That is the only unique thing about these lymphomas and leukemias. We also have cancer of the germ cells. The germ cells can also be mutated. So we have pluripotent cells having mutations. And an example is the cells in our testicles and our ovaries. So we have seminoma and then we have the other. We also have blastoma. And this developed from immature precursors or embryonic cells. And it is more common in children because as a child, you have your cells multiplying a lot, proliferating. And so you end up developing this type of cancer. We have other cancers like cancer in the brain, in the spinal cord, gliomas, and then we have endocrine cancers. These are all types of cancers, and you can pick any of these type of cancers to research on. You can pick any of them, whether you want carcinoma, sarcoma, leukemia, you can take any of these cancers. Okay, so we go to cancer immunotherapy. What has been done about the development of these cancers? So we have 6 types. I will only present 6 types. If we have others, I am not aware of, but then I know of 6 types of this cancer immunotherapy. So the first one I would like to talk about is the immune checkpoint inhibitors. So when a foreign antigen is introduced into the body, what happens is that immune reaction starts. They try to eliminate this foreign antigen. Now, when this foreign antigen has successfully been eliminated, what happens? There is still an infiltration of these immune cells. And so we have proteins that binds to these immune cells just to stop their activity. And these are the immune checkpoints. So the first type of cancer immunotherapy are these immune checkpoint inhibitors. So in cancer, in any type of cancer, when immune cells are initiated, when there is a shoot of these immune cells, cancer cells specifically produce these immune proteins to bind to these immune cells that has been initiated just to stop their activity. So these cancer cells are quite trickish that they know the immune cells will attack them. So they produce these proteins just to stop the immune cells before they even start their work. And so scientists sat down and said, if we develop inhibitors to these proteins, it can stop these cancer cells from multiplying. They wouldn't be able to evade the immune system. The immune system will carry on with their duty. And that is how come they came about with this immune checkpoint inhibitors. Now, the second one is the CAR T therapy, CAR T cell therapy. This is very common. And it has been utilized a lot because we find T cells as the major cells of adaptive immune system. They have the strong cytotoxic effects towards foreign antigens. And so this has been utilized a lot. So patients, what is done in this is that they isolate T cells from patients who already have this type of cancer. And as they isolate the T cells, they do some things, some skirmishes in the lab, and then add a chimeric antigen receptor, a receptor that specifically recognizes the cancer cells and eliminates them. So as they do that in the lab, they reintroduce these CAR T cells back to the patient because it's from the patient. And so these CAR T cells target the cancer cells and eliminate them. It has been successful with leukemias and lymphomas because of the circulation in blood. So you have availability of these cells a lot in blood. They last longer in the blood system. But with solid tumors that develop in mesenchymal cells in the bone or in muscles, there are challenges with penetration, how it will get to the tumor site. So that is CAR T cell. We also have cancer vaccines. So the function, it is just like the regular vaccines that we have. You introduce the body with a mutated form of the cancer just to initiate an immune reaction. And as you initiate it, it gets used to it. It already recognizes this foreign antigen, and then it fights it when it comes. We have cytokine therapy. So there are cytokines that boost immune activity. As we go on, you will see that there are other cytokines that also inhibit immune activity. So these cytokines are the ones that boost immune activity. So researchers thought of it and they said, okay, this can also be a type of cancer immunotherapy. The fifth one is oncolytic viruses. Oncolytic viruses, it just comes as nanoparticles. So typically viruses are coated with something, a nanoparticle. It can be any nanoparticle. So that these nanoparticles will guide the viruses just to arrive at the tumor site. And when it gets to the tumor site, the virus penetrates into tumor cells, they start to multiply and then cause the tumor cells to burst. And as they burst, viral antigens and then the tumor remnants are mopped up by T cells. So that is the rationale behind oncolytic viruses. So they use viruses just to cure tumors. A typical example is seen in glioblastoma. Glioblastoma happens in the brain. So these oncolytic viruses enters into the brain, cause the tumor cells to rupture, and then immune cells mop up the remnant. The sixth one, which is the last one, is adaptive cell transfer. So immune cells are collected. It can be any cell, but not T cells. It can be any cell. They are collected, enhanced, and expanded in the lab, and then reintroduced back to the patient to attack that specific cancer. Now we go to the specific osteosarcoma. No, sarcoma. Sarcoma first before osteosarcoma. So we already know it happens in mesenchymal cells, in connective tissues. We have angiosarcoma in the vessel walls, the blood vessel walls. We have osteosarcoma in the bone, leiomyosarcoma in the uterus, Ewing sarcoma also in the bone. We have fibrosarcoma, liposarcoma in fat cells. We have gastrointestinal stromal tumor. We have synovial sarcoma, rhabdomyosarcoma, and then the last one, you can mention it for yourself. So these are all tumors that are classified as sarcomas because they occur in mesenchymal cells of connective tissues. Now I decided to take osteosarcoma. This is because osteosarcoma happens a lot in children than in adults. If we have our children to be invalids, people that have illnesses, there wouldn't be an adult generation. So I decided to go in for osteosarcoma just because it is related much to the pediatric than in adults. It is easy to actually deal with children than to deal with adults. So osteosarcoma, a cancer of the bone. We have 3 cancers of the bone. How do I know this one is osteosarcoma? So osteosarcoma, we have it arising within the bone and bulging out, as you see in the picture. We have chondrosarcoma, which is also a sarcoma of the bone. We have it like presenting as necrosis within the bone, but somehow the morphology of the bone is maintained. And we have Ewing's sarcoma, which actually attaches to the bone. But then it doesn't eat into the bone as osteosarcoma, as you can see in the picture. Now, osteosarcoma, what is it? How does it come by? If we know how a certain cancer arises, we are able to prevent it. If you know how you fell sick, you are able to stay away from the how. But in this case, it is very difficult to know how it arises. If I get osteosarcoma, it can be different from another person who has also acquired osteosarcoma. So my osteosarcoma will be different from another person's osteosarcoma, and the other person's will be different from the other person. We can have 50 patients, and you will have different types of mutations in all of them. So it arises from bone tissues, the cells of the bone. The cells of the bone. And in most cases, you see them in children than in adults. But if you had it in childhood and by some means it went away, it is likely that you can have relapse above the age of 50. So above the age of 50, it is likely that you can have relapse. As I said, it has many inherited germline mutations, very different. Each person's osteosarcoma is just different. And one of the mutations include the TP53 gene. So basically, cancers arise as a result of a mutation in a gene that is responsible for cell growth, or a mutation in a gene that suppresses the signals that induce cell growth. So cancer actually is a multiplication of cells. You have your cells multiplying in excess. And even if they do that and they are functional, you are lucky. These cells multiply and they are not functional. They don't perform the natural process or the natural things that healthy cells are supposed to do. So if you get cancer, it's either the gene that suppresses multiplication has been eliminated, or the signal that suppresses the gene that causes that controls replication there is a problem with it. So we see the mutations, and this is the molecular perspective. So we see that we have signals that control cell division. The first signal, the WNT beta-catenin, it is responsible for proliferation and plasticity. When there is an alteration in this gene, there is excess proliferation and there is also excess plasticity. We see the PI3K-AKT. It regulates cell growth, survival, and metabolism. If there is a mutation in this signal, you have excess cell growth, survival, and metabolism. The cell tends to survive without apoptosis. We have the MAC, the RAS, RAF, MEK, ERK pathway. This pathway controls cell proliferation and differentiation. When there is a mutation, there is a problem. We have the NFKB, which suppresses apoptosis and is involved in inflammatory response. We have Notch. So when there is an alteration in these signals, you end up having excess proliferation. These signals, the mutations in these signals can differ from each individual. You can have one individual presenting with all these mutations. You can also have one individual presenting with only one mutation. And this, it all leads to the same osteosarcoma. Yes. So we go to the next one, which involves the actual genes. The most frequently mutated gene, which is the TP53. So most osteosarcoma presents with a mutation in TP53, which is responsible for apoptosis. So this is the most frequent mutated gene, the TP53. We have the RB1. Mutations in this tumor suppressor gene are frequently found in pediatric patients with osteosarcoma. So there can be a patient presenting with only TP53 mutation and another patient presenting with only RB1 mutation. It differs from individual to individual. Yes. We have the MDM2. So what this does is it negatively regulates tumor suppressor. As the numbers of MDM2 goes up, tumor suppressor goes down. The TP53 tumor suppressor goes down. As the numbers goes down, TP53 goes up. That is what it does. And cancer cells are able to express a lot of this gene just to hinder the effects of TP53. So you have TP53, but it wouldn't be able to perform the function you actually want. Another gene, the CMYC. So amplification of this gene, it is actually an oncogene. And when it is highly amplified, you have a lot of cancer cells. We also have other genes like NOTCH1, FAS, NF2, what. The actual focus is on immunotherapy. That is why I'm not emphasizing so much on these genes. So please permit me, okay? I forgot to mention one thing. So the picture on the left indicates, please, can you see my cursor moving?
Yes, we can.
Okay. So this is an actual osteosarcoma, which has started in a bone cell. And within this osteosarcoma, there are gene mutations. And within the same osteosarcoma, there is a microenvironment. And this microenvironment has tumor cells, it has cytokines, it has immune cells, it has growth factors, it has epithelial cells, it has extracellular matrix, and it has fibroblasts. It's like it has all the type of cells that are needed to fight an infection there. So this part is called the tumor microenvironment, the environment that surrounds the tumor, the environment in which the tumor actually develops. That is the tumor microenvironment. So let's look at it. If we are able to understand what is actually within the tumor, we will know how to manipulate our ways just to save lives. So this is a complex and a dynamic system of interacting cells. Each cell in this environment interacts with each other. And we also have structures surrounding the tumor. So structures like blood vessels, structures like collagen, structures like extracellular matrix. It is also a structure because it also forms part of the tumor microenvironment. So we have all that's in there. We will look at each component one after the other, and we will know where the exact research is supposed to be, what we need to do. So all these cells are able, they come in place to perform their functions. T cells come to perform their functions. Dendritic cells also perform their functions. Each cell represents something. They are performing their roles, as in how they will attack any foreign antigen. Okay, so we start with immune cells. We have immune cells, non-immune cells. We have extracellular matrix. When all these come together, they perform two functions. It is either they are suppressing the tumor or they are helping the tumor to suppress them. So they have two functions in the tumor microenvironment. They come into play to help us eliminate the cancer, or they help the cancer to eliminate them. That is their function. So the T cells, which are major cytotoxic cells, we have T helper cells, the helper CD4 cells, and cytotoxic CD8 cells. The helper CD4 cells perform effector functions actually. They also have ways of eliminating cancer cells, but the main function or the main reason for being at the site of the tumor is to stimulate CD8 cells to fight the tumor, because CD8 cells has the potential to perform serial killing. They kill one tumor cell and then goes to the other tumor and then kills it, goes to the other and then kills it. Yes, so they stimulate the CD8 cells to perform this function. The CD8 cells do this by releasing granules just to kill infected cells. What actually happens to T cells? So when I first encountered the tumor microenvironment and saw that there are immune cells, there are non-immune cells, what I actually thought of was, so if immune cells are able to infiltrate into this tumor site, why do we still need a cure? Because as time goes on, these immune cells will naturally eliminate the tumor. But it doesn't happen like that. There are challenges. So this is what actually happens to T cells at the tumor microenvironment, at the site of the tumor. So the effectiveness of CD8 cells is often hindered by immunosuppressive factors in the tumor microenvironment. And what are these immunosuppressive factors? They are other cells releasing cytokines. So these cytokines perform inhibitory actions. Instead of helping the CD8 cells, they rather inhibit cytokines, which makes them exhausted. So in a typical osteosarcoma sample, you might see CD8 cells, but you will see them exhausted, like they have no functionality. Their functions has been taken away from them. It has been ripped away from them by these cytokines. We already saw the checkpoints, immune checkpoints, PDL1 and PD1-CTLA. We already saw them. So what they do is to stop immune cells from acting excessively so that they don't cause autoimmune reactions. And so these cancer cells have the ability to produce excesses of these checkpoint cells, checkpoint antigens, checkpoint proteins. So as they produce them, they bind to T cells and then they stop the activity. So T cells are present, but then they cannot actually perform the function that they are supposed to perform just because they have lost their functionality. This is what happens to T cells at the site of the tumor. Now we go to regulatory T cells. We call them TREX. It is also a type of T cell as CD4, CD8. We also have TREX. Regulatory T cells because they are responsible for homeostasis at the tumor site. So they think we have to maintain a balance so that immune actions are not overexceeded. And so they come in to also perform their role. They play a suppressive role. So I am coming to stop immune action, but this immune action it is not even effective. One, the infiltration of these T cells to the point of the tumor has already been inhibited. Another one is cancer cells are already producing proteins that makes T cells exhausted. These T regulatory cells, on the other hand, do not know what is happening. And so they also release their cytokines that stop the activity of T cells, thinking that there can be an excess immune response. And so they need to stop them. And so they end up playing a suppressive role in the tumor microenvironment. So TREX can hinder anti-tumor immune response in cancer environment by suppressing cytotoxic T cells and other immune cells, not only T cells, but other immune cells. And as they do this, they promote immune evasion. So the only helpful cell that we've come across so far is the T cell. This regulatory T cells, we have already established that it performs a suppressive role. We go to NK cells, which is also a type of immune cell. So these NK cells, just as the T cells, also help with cancer elimination. So they also perform cytotoxic roles. They eliminate cancer, even without prior sensitization. So they go about checking which cell is healthy, which cell is not healthy, which cell should I eliminate, which one is supposed to stay. And so they end up eliminating those that they sense, no, this one doesn't present with MHC class 1, MHC, this one, and so they eliminate them. So NK cells can eliminate cancer cells through complex mechanisms. One, they release cytotoxic granules containing porphyrin, granzymes, granulysin, cytokines, and these cytokines, we have interferon gamma, we have the tumor necrosis factor. They produce these cytokines just to eliminate cancer cells. And then we see B cells. So B cells, they have dual roles. As we have TREX, we also have B regulatory cells, regulatory B cells. We also have them, and they also perform suppressive roles as well. They stop the activity of these B cells. So we have B cells presenting with suppression or promoting the tumor. So with suppressing the tumor, they produce antibodies and cytokines. They also present antigens to T cells, and they influence the action of other immune cells. So they have the ability to also present antigens to dendritic cells, so that dendritic cells can present to T cells for enhanced antigen presentation. So if I need T cells to do something, and then I send one person, go and tell him to do this. If another person goes, the T cell will see the severity of what I'm talking about, and so it will have an enhanced activity. So this is what the B cell does in enhancing the elimination of the tumor. So they primarily present tumor antigens to T cells and also stimulate dendritic cells to present antigens as well. The B cells also they have the ability to promote tumor. They secrete immunosuppressive cytokines such as interleukin-10, tumor growth factor beta, and they express regulatory molecules such as FASL and CD1D50. So these are also inhibitory proteins that stops the immune activity. We have B regulatory cells, which also promote the accumulation of TREX. So these B regulatory cells promote TREX, which we have already seen that it stops immune activity. And as it does that, it inhibits CD4 T cells and cytotoxic T lymphocytes, weakening the anti-tumor immune response needed to fight osteosarcoma. So B cells have dual responsibility in the immune, in the osteosarcoma tumor microenvironment. Now we come to dendritic cells. dendritic cells, they are basically antigen-presenting cells. So as they present their antigens, they are supposed to just go away. But no, they stay and do something else. There is a dynamic interplay where B cells can transfer antigens. I already mentioned this. But this, another role of these dendritic cells is to recruit TREX, which we already know has immunosuppressive activity, leading to immunosuppressive microenvironment. Once TREX come in, we know there will be an immunosuppressive environment. Now we go to non-immune cells. Non-immune cells, we have the tumor-associated macrophages. We have two types. We have M1 and then M2, depending on the activation state. If you are lucky to have excess of M1, there will be tumor suppression. You have your body's immune system fighting against the tumor. But if you're unlucky to have M2 in excess, you have poor prognosis because these M2 enhance immunosuppressive activities. So M1 macrophages are helpful, whilst M2 presents with immunosuppressive properties. We have cancer-associated fibroblasts, and the main function of these ones is to help with extracellular matrix, forming collagen and so forth. But in here, they promote the differentiation of M2 polarized macrophages, which present with poor prognosis. These promote tumor. They promote tumor growth. And so as they help with the differentiation of M2, you will definitely have poor prognosis. So CAFs and M2 macrophages cooperate to activate endothelial cells and their precursors, leading to the formation of new blood vessels, angiogenesis. So angiogenesis should have been a way that immune cells will be able to reach the tumor site. But unfortunately, when angiogenesis arises, the tumor becomes so dense and compact. So already it is within the bone and you have blood vessels diving into the bone. It becomes so compact. And so you have immune cells, but then because they are compact, sometimes they can't even travel to reach the tumor cells. So that is what angiogenesis does. If you have angiogenesis, a lot of that appearing in your sarcoma, it means it represents poor prognosis. So the table here is a summary of all that I've talked about. This is the tumor microenvironment. We have B cells presenting antigen to T cells. We also have them producing immunoglobulins. We also have T cells infiltrating into the tumor site. The CD4 helping CD8 to perform cytotoxic functions. We have chemokines released by B cells, released by dendritic cells, released by all types of the cells, also causing either immune inhibition or immune suppression. Did I say immune suppression? Immune inhibition or cancer proliferation. We have monocytes. Monocytes differentiating into macrophages, which we have M1 and M2. We have tumor-associated macrophages. These are the M1 and M2. Cancer-associated fibroblasts, enhancing the maturation of M2 cells. We have dendritic cells presenting antigens also to T cells. We have cytokines performing two functions, interleukin-6 and tumor necrotic factor, suppressing immunity by T cells. We have interleukin-2 and interferon gamma, improving immunity by T cells. And this, of course, represents cancer cell. So we go to the cure. So as I said previously, we were using surgery. I say we, clinicians were using surgery, typically using surgery. But they realized that there is a 15 to 20% of a 5-year survival. After 5 years, only 15 to 20% presents with disease free or survive it. And so they realized surgery alone cannot help because there are a lot of cancer cells that are left in the body. And so they added chemotherapy. And we saw as time went on, they realized that, well, chemotherapy is also helping, that there are also challenges because some of the chemotherapeutic agents present with cardiotoxicity. They affect other organs as well. And some of the chemotherapeutic agents, they are not even effective. As they enter the tumor site, these cytokines and proteins and all that are being released into the tumor microenvironment naturally eliminates them. So they are ineffective. And also they realize that when you have a relapse, it is difficult to treat because we've already performed surgery. We've used chemotherapy. It went away. And later you've had relapse. What again should we use? And so they went to radiotherapy. Now they decided to target these cancer cells with radiation. And they realized it is also causing a lot of harm rather than good. So they now thought of inventing a method which is safer. If I'm using your own immune system to treat yourself, it is safer than to introduce a foreign chemical or a foreign radiation or a blade to cut or something. And so they came about with immunotherapy. Now let's look at chemotherapy in osteosarcoma, specifically osteosarcoma. So the most used drug in this chemotherapy for osteosarcoma has always been doxorubicin. So in 18 something, they introduced doxorubicin, and it was helpful. It was quite helpful. But doxorubicin also presented with its own challenges. We will look at them. So it utilized the immune system to kill cancer cells. That is what doxorubicin does. One, it triggers CD8 T cells responses. So we all know CD8 T cells, they have cytotoxic effect. And so this doxorubicin triggers their activity. It also enhances the maturation of antigen-presenting cells, macrophages, and dendritic cells. We all know the function of macrophages in the tumor microenvironment of osteosarcoma. It has two main functions I already mentioned. Dendritic cells, we also saw that. So as it increases macrophages and dendritic cells, it also increases another aspect of immune inhibition. It is enhancing T cells, all right, but it is producing cells that inhibit T cells. So doxorubicin, let's look at doxorubicin. What is this about? It is an anthracycline. It has both cytostatic and cytotoxic actions. That is why it's able to introduce dendritic cells. It has direct membrane effect. This means that it is able to intercalate into DNA. It is able to penetrate into the cell and then inhibit topoisomerase 2. It is also able to disrupt mitochondrial function. When the powerhouse of a cell is shut down, it means the cell will die naturally. So it has that ability to shut down the powerhouse of the cell. It also has the ability to potentiate free radical generation and oxidative damage. Now, they realized that doxorubicin is also presenting with cardiotoxicity. It is toxic to the cells of the heart. And so they decided to use that in minimal quantities. So after surgery, they will now stick to doxorubicin and they will use them in minimal quantities so that it doesn't destroy your heart. In solving one problem, you end up creating another. And the decision that they made also made some cancer cells survive. If you don't use in excess, cancer cells, they survive. And how they survive, cancer cells has the ability to even go into hibernation. They will hide in other cells. They will hide somewhere that this doxorubicin cannot even reach them. And so as time goes on, you have osteosarcoma coming back. So some cancer cells were left alive. Now, they decided to see exactly what happens with the chemotherapeutic agent doxorubicin and the immune cells. If we actually know what is happening, we know how to help. So researchers decided to analyze what is the relationship between doxorubicin and the immune cells and non-immune cells. What relationship do they have so that we know what to do next? Now for T cells, they realize they don't have any problem because it activates CD4 and CD8 T cells. These cells are able to withstand cytotoxic effects of doxorubicin naturally. They also realized that doxorubicin does not induce cytotoxicity in T cells, but rather enhance their functionality. But regulatory T cells secrete immunosuppressive cytokines, TREX. This is TREX. They secrete immunosuppressive cytokines, which upregulate immune checkpoints, shielding tumor cells from elimination. So they rather shield tumor cells from elimination by doxorubicin. That is what Trex does. Now they checked NK cells. They realized doxorubicin and NK cells interact in a very complex way. At large doses, doxorubicin was able to cause NK cells dysfunction or make tumor cells more susceptible to NK-mediated destruction. So at large doses, they cause NK cell dysfunction. But then at minimal doses, they make tumor cells more susceptible to NK-mediated destruction. That is what this means. Dendritic cells were also looked into, and they realized they aid NK cell development, and doxorubicin can impede dendritic cell maturation. It also has that ability. I think it was at high doses. It is able to impede dendritic cell maturation. Okay, so the ones you see here, they are all found in my review paper, but I would like to read them to you just for the sake of this presentation. So we have NK cells and dendritic cells having a unique relationship. So each cell type regulates the other's maturation and function to coordinate immune responses against infections and tumors. We have dendritic cells activating resting NK cells, promoting them to kill target cells, while activated NK cells can also eliminate immature dendritic cells and promote their maturation. So this is the relationship between NK cells and dendritic cells. Now we go to macrophages. What does macrophages does when doxorubicin, DOX, has been introduced? So this one increased DOX resistance. What do? Within the tumor microenvironment, they provide an immunosuppressive pro-survivor milieu. Macrophages has that ability to phagocyte phagocytosing cancer cells, they rather phagocytose doxorubicin and reduce the amount of medication that reaches cancer cells. So they are there. Macrophages are there performing their normal function as phagocytosis, eliminating the cancer cells. Immediately DOX is introduced, they try to eliminate DOX. They take away DOX instead of the cancer cells. And by so doing, they cause cancer proliferation, even in the presence of DOX. DOX is not able to reach out to the cancer cells to eliminate them. Now we have cancer-associated fibroblasts. They cause DOX resistance by creating an excess of extracellular matrix element. So they produce collagen, and this collagen makes the tumor so dense that doxorubicin cannot actually penetrate. They cannot actually penetrate into the tumor core to kill the cancer cells. Okay, so as we have already established all these, now there comes immunotherapy. What should we target? Which of the cells do we target? Which of the effects of these cells should we maximize so that we don't find ourselves in trouble, so that we have a safe and a cancer-free state? CAR T cell therapy has already been established. There are, I think, there are methods and there are drugs available. CAR T cell therapy, but this is mostly used for lymphomas and leukemias, blood cancers. We call them blood cancers because they circulate in the blood system. You don't need to cross any barrier. You don't need to break any bone. So they are actually out and they are effective. We also have CAR NK cell therapy, which we have some of the drugs in clinical trials. It has also been effective. But the pure research ones are the B cells monoclonal antibodies. So B cells produce antibodies. These antibodies are helpful. So the scientists want to research into this. They actually want to research into this, know the type of antibodies that can be produced that is able to eliminate the disease. It is under research. You can choose that as also a research area. We also have antigen-presenting cells, dendritic cell vaccines. So these are dendritic cell vaccines. I don't know much about the dendritic cell vaccines because it's not my area, but then scientists are still researching into that. If anyone wants, they can also follow up that direction. We have suppressive myeloid cells, macrophages, myeloid-derived suppressor cells, which is also under research, if anybody wants that area. But these are the areas of research. These are the targeted therapies that researchers want to actually know more about. We go to the CAR T cell therapy. So this is a personalized immunotherapy. As I already said from the beginning, your cells, your own T cells are taken. They take it to the lab. They add a chimeric antigen receptor that targets the specific cancer. It targets a specific receptor on the cancer you are on osteosarcoma. This is related to osteosarcoma. So they grow osteosarcoma cells, and then they also grow T cells. They do some things, add this specific receptor to it, try it outside ex vivo, and then they now introduce it back into your system. It has been successful for blood cancers. For blood cancers, there is no point of inhibition. It has actually been successful. A lot of people are cancer-free. Those that had blood cancers, they are cancer-free because it is actually simple with the CAR T therapy. But then On solid tumors, the point of entry, how these T cells are able to even get to the tumor site is a challenge, and that is why it is under research. So approved uses, we have CAR T cell therapy, which is primarily approved for blood cancers. We have B cell acute lymphoblastic leukemia. It has been successful. Diffuse large B cell lymphoma. It has been successful. Follicular lymphoma, mantle cell lymphoma, multiple myeloma. It has all been successful because these are blood cancers. But then, particularly effective for blood cancers due to the nature of the T cells, as I said, because they are diffused in the blood system, there is no hindrances, there are no challenges. But then here comes the challenge. But then here comes the challenge. The immunosuppressive tumor microenvironment. Already we have things that are fighting the immune system. We have systems that are actually producing cytokines, chemokines, just to stop immune activity. So the immune system is already suppressed. It is suppressed in the tumor microenvironment. So that suppressive state, that is the problem. So we have solid tumors presenting with a dense physical barrier already in the bone. There is angiogenesis, there is collagen production. Already it is compact, abnormal vasculature, abundance of immunosuppressive cells and factors. They all hinder the CAR T cell infiltration and activation. So you will have it all right, but introducing it into the body is the problem. We have tumor heterogeneity. Now, what this is about is that my osteosarcoma is different from yours. So we will develop a CAR T cell thinking it will target this receptor or it would do this. But no, it is actually different. It might not even target it. So there is a widespread of heterogeneity of tumor antigen suppression, and this can lead to target escape, and it can also result in incomplete treatment or recurrence. And this heterogeneity, you will see genetic mutations which are different, protein expression on these cancer cells which are different, the cellular morphology different, cellular metabolism of tumor cells different, proliferation different, metastasis potential different in different individuals. And that is heterogeneity. We also have limited T cell persistence. So you have the T cells all right, but they wouldn't persist in the tumor microenvironment, leading to functional exhaustion. We have treatment-related toxicity. So CAR T cell therapy can cause treatment-related toxicity, which may limit its clinical application. That is graft versus host disease. Sometimes it happens. We take away your own T cell, we add this antigen, and by the time we reintroduce it back, your body is already rejecting it, is already pushing it out. These are all challenges. So the research part here is that all these challenges necessitate the development of innovative strategies such as cytokine armored CAR T cells. This will make the T cell last longer or even reach its site. We have logic gated systems and localized delivery approaches to overcome the limitation of CAR T cell therapy in solid tumors. Now we talk about the second already established therapy. That is the CAR NK cell therapy. This is also the same as the T cell therapy, but then NK cells can be derived from different individuals. It can be derived from umbilical cord blood, not necessarily from the patient who is suffering from the osteosarcoma. You can derive these NK cells from different individuals. Yes, pluripotent cells from different individuals, because you wouldn't have this graft versus host disease reaction. So as T cells, you have to isolate from the person suffering from osteosarcoma. NK cells, you need not to necessarily isolate from the person suffering from osteosarcoma. You can isolate from different individuals, and it can be prepared before, prior to treating the individual. You will have NK cell ready. But for T cells, you only have to get the disease before they extract your T cells to do this chimeric antigen receptor thing. But for NK cells, you will have the therapy already there. It is already there. You can have it already there because we don't have to necessarily take it from you. So just as the T cells, NK cells are modified with a chimeric antigen receptor. And the uniqueness of this is that as T cells recognize specific receptors, NK cells are even better. So we have some cancer cells also behaving as normal cells. So when T cells see a healthy cell that has major histocompatibility class 1, it will spare the cell. But NK cell will scrutinize the cell to see and confirm that indeed this is a healthy cell. So they are able to, some cancer cells present with the antigens that normal cells comes with. So these NK cells, the CAR NK cells, are able to figure out these cancer cells that are mimicking healthy cells. And that is the missing self signal. So T cell will recognize a self signal, but there are cancer cells with missing self. They don't pose as cancer cells, they pose as healthy cells. NK cells are able to take them out. Okay, so advantages of NK cells over the CAR T cells, as I already said, reduce risk of severe side effects, such as the graft versus host disease. There is also off-shelf availability. I already mentioned that we have allogenic use without extensive genetic modification. Just as T cells, you go through a lot of genetic modification and all that. NK cells are not like that. We also have rapid deployment. So unlike the CAR T cells, NK cells can be administered quickly, which is critical for aggressive cancers. It's not the case that you take the patient's cell and do something. It takes longer. These are readily available. So unlike CAR T cells, the CAR NK cells retain their innate cytotoxicity, including antibody-dependent cellular cytotoxicity and direct lysis of abnormal cells, providing a dual mechanism of tumor killing. Let's see the successes of NK cells. The application of NK cells has shown promising results in clinical trials, particularly in hematological malignancies, and these are blood cancers, leukemia, lymphoma. Yes, they have shown successes. So recent studies indicate that the CAR NK cell therapy has demonstrated safety and clinical activity, with significant therapeutic responses observed in patients. But there are also challenges. Challenges remain, including the need for further research to enhance their effectiveness in solid tumors and to improve their persistence in the body. It does not also persist in the body. Yes, because there is a dense tumor microenvironment. So there needs to be research on that, how it can persist in the body. So another key challenge There is limited in vivo persistence of NK cells. We already know. There is metabolic constraints and exhaustion induced by tumor immunosuppressive mechanisms. We also need to optimize the chimeric antigen receptor design for multiple antigen targeting and tumor microenvironment adaptation, how it will be able to live in that niche and not to only target one mutation. Okay. The focus of current research. So that was what I already mentioned. B-cell monoclonal antibodies still under research. You can actually choose that as your research area. Targeted pathways, the pathways that leads to immune suppression or immune activation. We also have antigen-presenting cells, the dendritic cell vaccines, and we also have the suppressive myeloid cells. All these are under research. It is still under research, and there are a lot of research papers you can find on them. Now, I would like to talk to you about what I am doing, which one I chose, and the direction of my research. So this is a review paper I published in May. Yes, it talks about everything I've talked about previously. This is just the abstract. So it talks about everything that I've said previously. If you have time to read about it, you will understand all of them. Yeah, all that I've talked about previously. All those are in here. Now, my area, what I'm actually doing. Oh, yes. So my research topic revolves around the development and production of a microfluidic platform for assessing metabolically active T cells and B cells in tumor tissues of patients with osteosarcoma. So I just want to know, there is a lot of exhaustion that happens with the T cell. When these T cells get to the tumor site, they are already exhausted. They are not functional. I want to know how many functional cells are present at the tumor site. And if indeed there are functional cells present at the tumor site, I'll now find out the type of these cells that are present there, and maybe how they even ended up there. So I already emphasized on osteosarcoma. We already know it happens a lot in children than in adults. And first, they were using chemotherapy and surgery. Chemotherapy, Dr. Rubison, we've talked about it. It has been used over the years, and they now thought that there can be a safe method, an effective method, and so they introduced immunotherapy. Now, my goal is to estimate the numbers of T cells and B cells present at the tumor sites. This one, whether active cells or inactive cells, I just want to know, are there T cells at all, or are there B cells at all? Because this microenvironment is already dense. Infiltration of these immune cells is limited. So I want to know, are there T cells at all? I want to also see the correlation and the numbers of these immune cells to the severity of the disease. So if there are T cells or there are B cells and they are functional, to what extent is the disease? How bad is the diseases? Okay. I will also correlate the numbers of these cells to recurrence. So we have patients that had osteosarcoma in the past and they went away, but now they are having recurrence. So I want to see. I cannot have the same patient coming in with a primary osteosarcoma, it went away and it came. I cannot compare with the same patient, but at least I can compare with a different person, first timer, and then a recurrent patient. And that is one of my goals. I want to also evaluate the effectiveness of anti-tumor therapy based on the analysis of these immune cells. So if indeed there are T cells that are viable or there are B cells that are viable, and I check the severity of the disease and I see that, oh, okay, because these cells are viable, indeed it is like this, that is when I'll make a conclusion. Okay, so these are my tasks to compare the population and composition of intracellular lymphocytes in primary and recurrent tissues of osteosarcoma. I also want to evaluate the metabolic activity of intracellular lymphocytes using a microfluidic chamber. So microfluidic chamber helps us to isolate viable cells, cells that are actually active. And that is why there is an introduction of this microfluidic chamber. So, sorry, some of the words are in Russian language. Yes, I'm so sorry, some of them are in Russian language, but you just have to cope. I will explain everything. So, after the study is actually unique to determine the presence of T cells and B cells in the microenvironment of osteosarcoma tumors. I will isolate metabolically active T cells and B cells. I limited to T cells and B cells because I realized that T cells is actually dominant. You actually see T cells performing most of the functions, and that was why I limited to T cells. And then B cells, well, even though they have dual functionalities, but I just want to see what happens actually with B cells. I also want to identify the subtypes of T cells and B cells in the microenvironment. So if I realize there is indeed viable T cells and they are indeed viable, and I check compared to the severity of the disease, and I realize that no, when there are viable T cells, there is a less severity of this disease. I want to know which type of T cell has been able to cross the barrier, which type of T cell has been able to succeed. And if I know the type of T cell, then it can also help with immunotherapy. I want to know the number and the usefulness of T cells and B cells as immunotherapeutic agents for osteosarcoma. Okay. So this is basically the theoretical significance, just to expand the research on immune cells in osteosarcoma treatment, particularly B cells and then T cells. So I also want to know if the increase in T cells and B cells activities can actually be considered as an important strategy for the elimination of osteosarcoma and with its recurrence. So osteosarcoma has a lot of heterogeneity. Yes, we already have this theoretical background about T cells and B cells and all that. Is it actually the T cells that helps or B cells? Because it hasn't been established yet. We've seen the theoretical part, but then what actually helps with healing? That is this point. I also want to provide a clear understanding of the increased activity of immune cells and good prognosis in osteosarcoma. This is the practical significance to provide a representative basis for immunotherapy and its essence in the treatment of osteosarcoma and its relapses. Also, to offer a potential strategy for the treatment of osteosarcoma, and also to help improve the treatment planning, prognosis, and survival of patients. My expected outcome is that I expect that the presence of these metabolically active T cells and B cells will ensure the suppression of the osteosarcoma cells. If it happens that way, then I've succeeded. If it doesn't, then research continues. I also expect that the numbers and the proportions of T cells and B cells will become a therapeutic tool to prevent the recurrence of osteosarcoma, the recurrence particularly. So actually, I'm dealing with 36 patients from oncology clinic in Russia. They are all kids with osteosarcoma. Actually, the hospital prepares these samples and they put it into blocks and they send it to my university. Yeah, there is that connection with them. So that is what happens. I'll be working on 36 patients for now. Well, I don't pray that people get osteosarcoma, but then I hope that I work with 36. And if in case anybody also gets, it will add to the numbers. So it can be 36 or more, depending on how many people get this cancer. Okay, so when I started, it was 33, but three more had cancers, and now it's 36. Yeah. I think I've already talked about this. Yes. So thank you so much, and thank you for your time.
Thank you very much, Augustina, for this wonderful presentation. I believe we've learned a lot. We've really delved into immunology and its application. It's really interesting. I'm really glad for this presentation. Please, the floor is open for questions. So that Augustina can answer. So please, the floor is open. Questions. Let me hear. I'm sure you have a lot of questions because she spoke a lot. Yeah.
Okay.
Yeah. Please ask your questions. Thank you. If you also want to put it in the chat box, you can do so and I'll read it out. But if you also want to just speak, you can just raise your hand. I'll call you and then you can ask the question. Thank you. Okay, so as we wait for the others to ask their question, listen, I was thinking, okay, Stephen, ask your question. Ask.
Yeah, thank you very much, Mr. Santua, for this wonderful presentation. I really enjoyed. But I have a question concerning the correlation between blood cancers and osteosarcoma. So, because osteosarcoma deals with cancer of the bone and leukemia actually starts from the bone marrow, have you gotten any, some of your patients, is there any relationship between leukemia and the osteosarcomas, or is any of your patients having leukemia? I don't know if I'm asking the right question, but I just want to know if people with osteosarcoma can also develop blood cancer, since this is a cancer of the bone and which can actually affect the bone marrow. Thank you very much.
Okay. You're welcome, Stephen. Actually, what the mutations are different. So the mutations in lymphomas and leukemias are different from that happens in osteosarcoma. But why not? Nobody is exceptional. That is what I would say. Nobody is exceptional. I haven't actually come across somebody suffering from lymphoma or leukemia and then osteosarcoma at the same time. Yes. But then it can be, you can have multiple cancers, and actually osteosarcoma can metastasize. So you will have osteosarcoma, and by the time you realize, you are presented with a lung cancer in the lungs, because it has metastasized. Yes, so there can be anything, but then I haven't come across any patient presenting with these two cancers at the same time. But what you have to know is that the mutations are different. So the mutations in osteosarcoma is different from the mutation in lymphoma and then leukemia. And the presentations are also different, even though one is in the bone And this one, the preparation starts in the bone and it's pumped out into the blood system. The mutations are different and their expressions are also different. But you can actually get these two cancers at the same time. Who knows? Yeah.
Thank you very much, Stephen, for your question, and thank you, Augustina.
I hope I answered his question.
I can see a thumbs up from his end, so I'm sure. Stephen, did she answer your question?
Oh, yes, I'm good. Yeah, thank you.
All right. Please, the floor is open for question. You can also put it in the chat box if you have a question. I was thinking, Christina,
Yeah.
pardon my ignorance as usual.
Uh-huh.
Osteosarcoma, the plasticity, is it epithelial mesenchymal or mesenchymal epithelial? And how does does immunotherapy has any kind of advantage over this kind of transition? Because yes, we are trying to get the immune system to swarm up to attack, but if the transition is also somewhere, will it have an effect? Maybe I'm just speaking things, but do I have a point? And can you?
You are mixing them up.
Uh-huh.
Mesenchymal cells. We have mesenchymal cells and then we have epithelial cells.
Yes.
If the cancer happens in epithelial cells, we call them carcinomas.
Yeah, I know that.
So if it happens in epithelial cell, it is termed as a carcinoma. But if it happens in a mesenchymal cell, in a connective tissue, it is called a sarcoma. So osteosarcoma happens in the connective tissue of the bone. So mesenchymal cells of the bone, that is where it happens.
Okay.
Please do, so it happens in the mesenchymal cell of the bone. So it happens within the bone, not the cells that lines any bone, muscle. No. It just happens inside the bone.
Okay. All right. Thank you for the clarification.
You're welcome.
Please, the floor is open for questions. So if you have a question, you can ask. Hey, please, you can't tell me you are okay. You kindly ask your questions.
It's a lot to, I think it's a lot to push in.
Eugene,
But
kindly
then
ask
as
your
you
question.
read,
Augustina, sorry, Eugene has a question.
no, okay, yeah.
Eugene, please ask your question if you can hear me. Eugene. I guess maybe it was a mistake. Eugene, your hand is raised. But Augustina, please, you can go on. You were saying something. Can go on.
Hey, I've forgotten.
Oh.
I was saying that as you read the review paper, yes, you will understand a lot of things, how the immune system acts, particularly in osteosarcoma. Yeah. But then for the first hearing, it's actually a lot to grab in. Yeah.
So please, I'm waiting for your questions. This one I was looking at. So please talk a little bit about the microfluid thing? No, I mean, I didn't really get that part.
The microfluid chamber. I
Yes.
actually mentioned it in past because I actually didn't want to bother you with a lot of things this evening. So actually the microfluid chamber, they use chips. It can be chips and field. It can be electromagnetic field. It can be chips coated with immune cells like T cells, or it can be the cells you want to actually isolate. But then it helps isolate viable cells, cells that are metabolically active, cells that are not exhausted. So this chamber will help us isolate cells that are actually active from the tumor microenvironment, from the osteosarcoma samples that we're gonna take. So it means that we don't need paraffin embedded samples. We actually need fresh samples from patients. Yes. And then we isolate these viable T cells and then estimate their metabolic activity. I was told that it involves a lot of money, but then we are still trying to see if it will still be successful or it will still be possible. Yeah. So basically, that is what it does. It actually isolates viable cells, cells that are metabolically active, that are not exhausted. Yeah.
Senior Oral, please, I have my question in the chat. Thank you.
Yes, I've seen it. Eugene is asking what the current immunotherapy interventions that's available in the industry. And in your research, are you thinking of starting any drug trials?
So there are a lot of clinical trials that is going on. Unfortunately, I did not put in the names of these drugs. I don't know if I actually did at the T cell type. Let me check. Because I saw a lot of them. I saw a lot of them there. So we have a lot of these T cells available. Unfortunately, I did not put any of them there. We have brands that are already manufacturing these T cells, these CAR T cells, brands that are manufacturing them. And as I said, it has already been successful in these type of cancers, B cells, lymphoblastic leukemia. It has already been successful, but the challenge is solid tumors. That's the only challenge. Yes, and NK cells has also been successful, but in my case, I am trying to address another challenge altogether. After finding out which cells are viable, I want to see the composition of the cells. So we already know T cells help in eliminating the disease. Yes. But after finding out that, okay, there are viable cells in there, and these viable cells relate to a patient who is actually cooperating, like responding well to treatment. The patient has a good prognosis. Once I find that, I will now see the subtypes of these T cells, because it could be that the composition or the content of these T cells are actually helping with the disease, with a good prognosis. So I find the subtypes of these T cells present, and now I make a conclusion. I think it is a continuous research, so I wouldn't be the last to complete it. There might be somebody who will also follow up, after I'm done. But the goal is to develop a drug. That is the goal. My school is a research center, and then that is what they do. Yeah, they send a lot of drugs for clinical trials, and finally they develop these drugs and they start treating patients with them. So that is the final goal, to develop something that is able to help with relapse of osteosarcoma, the treatment of a primary tumor and its relapse. So that is the final goal.
Right. Thank you.
Okay.
Jude says that wonderful presentation, and he's hoping to see more of immunohistochemistry incorporated into this area of research. Yeah, that's fine.
Mm-hmm. Immunohistochemistry.
Yeah, immunohistochemistry.
So the first part that I said I wanted to see T cells and B cells, the presence of T cells and B cells at the tumor site. It is typically immunohistochemistry. So we are using immunohistochemistry methods to know these, to identify these T cells and B cells. And what we have done so far is that we've used antibodies to, we stained these samples, paraffinized blocks. We sliced them and then stained them with these antibodies, and we observed. But the first one with a normal microscope failed. We couldn't identify any T cell or B cell. We used a fluorescent microscope. It also failed. And then the last one we used was a confocal microscope, or I've forgotten the name, but this microscope uses laser. Yes, so we use that microscope to actually find out whether there is the presence of even T cells or B cells. And that is the stage we are now. It is purely immunohistochemistry. Yeah.
Oh, okay. Please, Jude, I hope she has answered your question. Right. He says thank you.
You're welcome.
Yeah. Eugene, please, do you still have a question? I can see your hand up. Right. I believe today we've learned a lot, and I must admit I'm very grateful, Augustina, for making the time. Please, I don't know what time it is in Russia. Please, what says the time now?
It's now 9:47.
9:47. Wow. Wow.
Yeah, in the night.
Thank you. Thank you very much for
You're
making the
welcome.
to speak to us. We hope to see you another time. Also, thank you very much, all participants, for making joining this webinar today. And this is the final episode for the second and third quarter expert series. Maybe later in the year we'll organize another set of series, and then with workshops included. Right. And so, Augustina, thank you so
Mm.
much. God richly bless you.
You
And
are welcome,
um,
Raj.
please
Yeah.
have a lovely evening. To the
Thank you
participants,
so much.
kindly also have a lovely evening and enjoy your weekend. God bless you all. At this moment, I'll just say bye-bye.
Okay. Goodbye.
Bye.